So as an overview for today's talk, I'm going to focus on the bottlenecks that we and most large-scale projects that are designed along these lines will invariably face. But it's not all doom and gloom. I'm going to highlight the ways we've surmounted most of these bottlenecks. So we'll talk about how one generates the data. This will be a review for most of you. Assembling the infrastructure to actually pull off a large-scale project that involves data collection, analysis, and delivery of results. And as a key part of that, the final bottleneck being managing and analyzing the data. And that's where the GNOME platform that we've adopted at UBC will come into play. Just a 30-second review of genetic variation for all of you in the audience who undoubtedly know better. The DNA replication machinery, thankfully, is extraordinarily accurate, but every once in a while, an error is made, and that error is the fuel of variation in evolution. And genetic variants can take a number of different forms. We're still discovering some of the types of variation in the forms of chromatripsis, et but for today, I'm going to focus on the single nucleotide polymorphisms and how we can use information about individuals' SNPs to hopefully guide drug therapy and avoid adverse drug reactions. So what can we hope to learn from studying SNPs? As I just alluded to, our primary goal, or one primary goal, is to identify SNPs that are associated with disease development. There are several large-scale projects in diabetes, heart disease, the association with variation and predisposition to addiction, as well as Alzheimer's and autism, and not to mention the numerous success stories with cancer genomics. One can also identify patients who will benefit from certain drugs. There are, in fact, drugs that cannot have any therapeutic benefit to certain genotypes, and so there's no utility giving patients those particular medications. And then finally, what our project is focusing on is being able to predict the differential response to drug therapies and using genotypic information to be able to adjust the dose of drugs to fit a particular patient's genotype. So as I said, I'm going to try to hit on 3 bottlenecks today. The first bottleneck historically has been to generate the data, and we all know the story of the Human Genome Project taking about 10 years, although the bulk of the sequencing was done in the last 2, and for one to a few individuals, the cost was $3 billion. And then the projections were that we should be near the $1,000 genome right about 2013 if the impetus from the National Human Genome Research Institute paid off. And in fact, it did. And right about now, we are at the $1,000 or so genome, depending upon how you do the math. And so I'm not going to look to the future and rather make the case that the time for translating human genotypic information into patient care is in fact now. And so we need to move on to addressing the downstream bottlenecks.